It's a real pleasure to be here. I'm not quite sure. I first came to Hungary in communist times when it was really quite difficult, and we had to go immediately from the airport to Szeged along a very long road with lots of trucks on it, and boy, how times have changed since then. I was reminded in the museum the day before yesterday, I went into the museum, what a grand place Hungary used to be in the Middle Ages. I mean, there are some incredible altarpieces there. I'll show you one thing. And it's such a beautiful city, too. It's really a pleasure to be here. This is the view from my bedroom window. People ask, has the Nobel Prize changed your life? And yes, it has. You get to stay in much nicer hotels. So I stupidly didn't write down the name of the artist or the title of the picture, and I really don't know what's going on here, but I thought they were sort of in a cycle, and this little looks like Hieronymus Bosch, but I don't think it is. There's an angel that I cut off. Isn't that wonderful and mysterious? I love that. So that's what science is all about, trying to figure out what's going on. That's all it is, actually. People say you have to publish papers and in high-impact factors, but those are only surrogates. What it's actually about is finding stuff out. Very simple. Discovering things. Now, Andras said that I was a professor. I've never been a professor. In my day, you see, the professor was the head of the department, right? He was the grand panjandrum. And I never aspired to that at all. Then it sort of grade inflation took over and everybody became a professor, but I always avoided becoming a professor, so I'm not really a professor. That's number one. Number two, I actually stopped running a lab a long time ago at the end of 2010, so I really am a living fossil. I shouldn't really be here at all, and what I'm talking about is stuff that I did a long time ago, and I don't really have enough time to explain it properly either. So please forgive me if, and maybe if somebody says, wait a minute, put up a hand or shout or something, and we'll see what we can do. So, the other thing I should warn you is that it turns out this is a practical thing for all of those of you to go out to give a talk. Every so often, you come to place and you set up your computer, right? And usually if you arrive 10 minutes or a quarter of an hour beforehand, it takes about 2 seconds to set everything up. But occasionally things don't work, and actually here things don't work. And the reason they don't work is that the lead from here to the sort of control center over there is too long for my computer to handshake properly with the projector, which is all the way up there. Now, this is electronic engineering, which I don't understand. But the solution is that my computer is now somewhere over there, and my little clicker has to talk to a radio receiver that's just hidden behind that loudspeaker. So I hope it works. And if it doesn't, if I wander too far over that way, I will get out of range and the slides won't move. So all this is important practical stuff. So let's see. These are some cells on a dish, and that's the first cell that's going to divide, that's the second cell that's going to start dividing, and that's a sort of third. Now I want to see what happens. See, it rounds up, divides in two. Rounds up, but wait a moment, it hasn't divided. Meanwhile, cell number 3 is divided. Number 2 is still there. What's going on? All the other cells, so my question is, what is happening? You can see that something very dramatic happens when the cell divides. They round up, and you can see it sort of jiggling inside. And I want to understand why and when and how and what's going on. Because this is pretty important. I'm now going to show you the early life of a fish. And it's composed of 2 parts. First of all, the fertilized egg is a huge thing in the case of a fish. And it has to divide many times for the cells to become small enough so that they can move and then become different and turn into nerve cells and muscle cells and skin cells and eye cells and so on. And this is a beautiful movie made by this girl in Heidelberg with some very fancy microscopy that I don't understand. But I really like this. I think so. So watch, the beginning is lots of cell division making lots of new cells, and then they start moving rather like ants, you'll see. So amazing, doing, 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 until you have about sort of 4,000 or 8,000 cells. And then the cell division really pretty much stops, and forming a fish mostly becomes a matter of moving around. You see, they really do look like ants, sort of crawling about. And the amazing thing is that all of these cells start out the same, and then they become different. And every single cell, just look at this, you begin to see that this is going to be the head of the fish up at the top, right, and the tail down at the bottom. And then, so, this is what biologists have to explain. So I'm right at the beginning, the cell division bit is relatively simple compared to the cells becoming different things. But the key thing is that every single one of those cells could, in principle, take the place of any one of the other cells, because all of the cells have a complete library, the library of DNA. And it's just a matter of, when you go to a library, which book do you take off the shelf? Well, it's in the DNA, which gene or set of genes do you express? So it's absolutely critical that every cell gets a complete copy of the DNA, because that means that every cell could, in principle, take the place of any other cell. Okay? So, in order for every cell to have the same DNA, there are two processes that have to go on absolutely faithfully. The first is that the DNA has to replicate. That's what's going on here. I mean, of course, this conceals tremendous complexity. We now know in considerable detail, but not perfect detail, how this actually happens. And it's terribly, terribly complicated, and it's not my thing. And then after the replication, we have to separate the two daughters into two daughter cells so that the process can repeat it. And that basically is the cell cycle. Now, in this cell cycle, it's terribly important that the right thing be done at the right time. And I'm going to show you that in just a second. And what interests me is turning things on and turning things off, and the switches. There's the biochemical hardware and software that bring these things about. And actually, in this cell cycle, there are really three important places where there are switches. And they all have this characteristic that they are points of no return. Once you've decided to go on, the decision is taken carefully, but once you take it, it's quite irrevocable. So the first one is getting into mitosis, which we just saw in that movie. The second one is getting out of mitosis, and we saw that one cell that didn't seem to be able to get out. Why couldn't it get out? Because it couldn't line up its chromosomes properly, and it knew that it couldn't line up its chromosomes properly. And until you pull the chromosomes apart, you can't, you've got to line up. Otherwise, the two daughter cells won't have the same complement of DNA. And then the third place, which is also very important, but it's just too complicated for my poor brain, is starting DNA replication. Obviously, once you've started replicating DNA, you can't decide that, oh my God, we're running out of phosphorus, or there's not enough nitrogen, or something like that. You can't then unknit the DNA that you've already made and go back. You have to plow on, although it may be a disaster. So the first time that anybody really sort of made an understanding into these switches is these wonderful experiments which were done by Bob Johnson and Paktu Rao many, many years ago. And technically, these were a marvel, because what they did was to synchronize cells at different phases of the cell cycle, M, G1, S, G2, mitosis, a gap, DNA synthesis, another gap, mitosis, okay? And then fuse them together, which they had a very neat way of doing, and ask what happens. Well, obviously, if you fuse a cell in G1 with a cell in G1, the heterokaryon, the fusion product, is in G1, and so on and so forth. But look what happens if you fuse an M-phase cell with a cell at any earlier stage in the cell cycle. They always enter mitosis. There is something in mitotic cells, actually those rounded up cells you could see the chromosomes jiggling inside, which is dominant over all other phases of the cell cycle. And that's sort of what I'm going to basically tell you about. Now, so that, let's see what that looks like in practice. So here, two cells have been fused, one cell in mitosis, and you can tell these are mitotic condensed chromosomes. If the companion cell is in G1 phase, of course the chromosomes haven't replicated yet, so you have single threads instead of double threads. Now that, of course, would be a disaster when it enters mitosis because you've got no chromosomes to separate. So, what happens? Disaster. And then what happens if you enter mitosis prematurely while you're still replicating the DNA? You get a phenomenon known as pulverization. It just looks terrible. And the reason is because you're trying to condense chromosomes that are in the act of replication. They're very sensitive, and actually, if you think about it, very easily broken. So actually, the chromosomes just get shredded. And I think this occasionally happens in cancer cells. It's a very good way of generating cells with horribly malformed chromosomes. So it's something to be avoided absolutely at all costs. There must be a very strong mechanism that stops that happening. If you fuse a G2 cell with an M phase cell, it's really not so bad. I mean, you can tell the G2 aren't quite as condensed, but that's... So the important thing then is that this decision to enter mitosis should be taken with extreme care. And actually, these experiments revealed that, because if you fuse a G2 cell with an M phase cell. Now let's see. Well, let's do a G1 phase cell with a G2 phase cell. So the G2 cell is, let's say, half an hour away from entering mitosis, but the G1 cell is maybe 18 hours away from entering mitosis. So you know what's going to happen. The answer is that the G1 cell has to first pass through S phase before it gets into G2, and the G2 nucleus or the G2 cytoplasm respects that. It somehow knows that it shouldn't be entering mitosis until replication has finished. And it may amaze you to know that we still don't really fully understand what that mechanism is. How does a cell, because if you think about it, in principle, even a single replication fork that is not completed should delay the entry into mitosis. And that would be an amazingly sensitive receptor mechanism. And it's still something that puzzles me. Interestingly, if you fuse an S phase cell with a G1 phase cell, that pulls the cell, the G1, it advances. So there is... Basically, as you go through the cell cycle, things become more and more dominant. It's usually dominant over the thing before. But this border here, entering mitosis, is a decision. Once taken, it's irrevocable and dominant, but it's taken with extreme care in real life. Okay. So, and then another thing which, in the movie you just see, you can't really see what's going on, but as those of you who've done elementary biology will know, mitosis is actually rather a complicated process. And in eukaryotic cells, it means that, for example, these poles which are going to make the mitotic spindle have to separate early on. Chromosomes start to condense, complete their condensation, and then suddenly the nuclear envelope breaks down and the cells would round up and the chromosomes line up on the metaphase plate. And then comes the most wonderful transition, the metaphase to anaphase transition. This is also a decision point. If the chromosomes are lined up, then you're allowed to do this in the cells. There's a delay while the two sets of chromosomes get nice and far apart. That's important. And then the cell divides in two. Okay, so it isn't really a sort of exactly, there are things unwind in time as well as just switch on and switch off. So how do we know about this? How do we find out about this? Well, the first great pioneer, really, in getting into the molecules was Lee Hartwell, who I'm not going to show you his experiments, but here's Paul Nurse, who followed in Lee's footsteps, and Paul was my boss. In some ways, I guess he still is, actually, because I have an affiliation at the Francis Crick Institute in London, although I don't really spend any time there these days. But Paul, following Lee's, isolated mutants that couldn't divide. So there was something wrong with the control process. So the CDC2 mutant at the non-permissive temperature could not enter mitosis. It lacked the thing that makes you enter mitosis. That thing was very commonly known in sort of biochemical circles as MPF, maturation promoting factor, or you could think of it as M-phase promoting factor. And basically, these CDC2 mutants don't have that. And then in trying to you can get lots of these different mutants defective in different parts of the switching machinery and defective in different parts of the DNA replication machinery and so on. Paul noticed that there were these cells which at a non-permissive temperature divised prematurely. And he called these Wee1 because he was working in Edinburgh at the time, and Wee is the Scottish for small. Okay? And he realized this was an extremely interesting mutation because it actually seemed to accelerate cell cycle progression rather than inhibit it as this one did. Okay? Now, my contribution to this was nobody knew how this CDC2 was turned on and off. And in fact, when CDC2 was finally sequenced and cloned, it seemed to encode a protein kinase, the enzyme that sticks phosphates onto other proteins, which we'll come back to in a second. But when you made this protein in bacteria, it had absolutely no activity. I remember walking around a field in Bath discussing this with Paul. And he said, you know, it's definitely not a protein kinase. I know it looks like one, but it isn't because we've tested, and it isn't a protein kinase. Well, there were two reasons why it wasn't a protein kinase in Paul's hand. One, it didn't have its activating subunit, and two, he used the wrong substrate anyway. So, I was just the lucky person. So I provided inadvertently, it took us years to figure this out, really terrible, embarrassing in retrospect. I discovered these proteins here, cyclins A and B, and you can see why I called them cyclins, because they came and went. These are disappearing proteins. I always found it very difficult. You know, how can you explain this to your grandmother? What did you win the prize for? Well, something went away. It doesn't sound right somehow. Disappeared. And it took us a very long time to figure out how it disappeared and how its disappearance was regulated. And you can already see, so these you know, it's pretty amazing. When I saw this for the first time, I kind of knew that I'd made a really important discovery, because at the time it would have been considered quite impossible for a protein to disappear like this. I mean, of course, proteins disappear in your stomach because there are enzymes, but everything disappears in your stomach. But how can just one protein or two proteins go away in a cell when all the other proteins are perfectly stable? So, you know, so this was nobody had even suggested this theoretically. But I worried, you know, could this really be true? Number one, these are radioactive proteins, actually. So can you really make enough? And the time scale here is about half an hour between divisions. Can you really make enough protein in half an hour to catalyze cell division? Because that would be the hypothesis. And second of all, you can see these proteins, of course, by their very nature, accumulate gradually, linearly. But entry into mitosis is something that happens rather abruptly. How do you convert this gradual increase in something into a sharp, switch-like transition? So these are two things that, you know, one shouldn't really have worried about. And then, of course, what triggers and what's the mechanism of its disappearance. But to cut a long story short, and it was a long story, about 4 or 5 years' work, what we discovered was that here on the right-hand side is cyclin, here is Paul Nurse's and Lee Hartwell's protein on the left-hand side. And the way this works is that this is the activating subunit. You see this loop of protein here. That sort of nestles in a nice fold on the surface of the cyclin. And without that, this folds back and obscures. You can't see the ATP because it's not present in this diagram. So it's really very, very simple. And that's sort of, in a way, why, you know, it's nice when things are really, really simple. Because all you have to do is just make cyclin, combines with the CDC2, turns on a protein kinase, phosphorylates lots of proteins, and you're in mitosis. Fantastic. And then somehow the cell degrades cyclin, that turns off CDC2, and the cell gets out of mitosis, right? Easy peasy, textbook story. But it's a little bit more complicated than that, because you remember Wee1. Well, it turned out I guess it was Cathy Gould who discovered that WEE1 phosphorylated tyrosine residue on CDC2 to stop it being active. And then there was an enzyme, so and this one, I mean, you know, this is complicated stuff. It has both an activating phosphate and an inactivating phosphate. And the inactivating phosphate is removed by a protein phosphatase called CDC25. So the balance of activity of these enzymes determines the activity of this. And then you have to have CDC13 as well. So, you know, it begins to look a little bit complicated. And then it gets really quite puzzling, actually, because it turned out that CDK1, that's the holoenzyme, cyclin plus CDC2, inhibits WEE1, and WEE1 inhibits. So in interphase, WEE1 is dominant, and you can't turn this on. In mitosis, this is dominant. And so how the hell do you switch? According to this mechanism, once you're in one state, you're stuck there forever. So this is really, you know, that's a really important problem, and not an easy one to resolve, actually. So this is where the Hungarian connection comes in. This is my friend Béla, who now, he deserted Hungary and went to work in Oxford, actually. And here you can see he's worrying about WEE1, and there are lots of other things. And together with an American colleague, John Tyson, they came up with a very nice model which explained the thing. But they took values for the activity of these enzymes, WEE1, CDC2, the level of cyclin, plugged them into differential equations, and claimed to be able to explain the cell cycle. So the theoretical biologists always love things that cycle, you know, circadian rhythms, things like that. And you can see their model, it really looks rather convincing. And you can see cyclin accumulates. I'm not quite sure why the curves in that. I would have it going straight up, but never mind, that's their model. And here's tyrosine 15, which inhibits CDC2. And you can see that as the cyclin comes on, you get more and more tyrosine. So that keeps that in check. So the MPF activity, which is going to get you into mitosis, is held in check until something happens at this point here, where the cyclin, I mean, maybe there's a little bit of cyclin activity, so you get a little bit of CDC2 activity, which then starts to turn off the WEE1. I think that's probably the easiest way of thinking about this. But everybody said this was the 1990s. Everyone said, oh, that's great, so now you've explained everything. But actually it didn't. And now I want to explain why that model is wrong. And it's experimentally wrong, too. And what this is where Béla says, you used to hate me, Tim, he says, because I was sort of skeptical about these modelers because they tended to only take the things you know and not suggest things that you didn't know. And I like it better when, you know, they tell you, wait a moment, you're missing components. And we were missing components, and I'm going to explain that very quickly because we're running out of time. So the basis of this is that we have a cell-free system, which is pure cytoplasm. And the cell-free system can nevertheless do cell division. You add nuclei to these cell-free, and here are the chromosomes, and here are these beautiful spindles. And if we do add some calcium, they would do the metaphase-anaphase transition. Now, using that system, it's possible then to ask, what do you need to get it to enter M phase? And this is a key experiment, really, done by Mark Solomon in Mark Kirshner's lab. Okay, so you add in some cyclin, and when you add a little bit of cyclin, although you think you might get some kinase, you don't, and the reason you don't is because it's held down. And then you exceed some threshold, we're just adding in extra cyclin here, and then suddenly it turns on. So what's the explanation for that? Well, there are two kinds of explanation, and this is where the switches come in. You know, you can think of a doorbell, which when you press it, as long as you stay pressing it, the bell will ring. But the minute you release the pressure, it stops ringing. And this switch, on the other hand, a rather more conventional light switch, is a bistable switch. If it's off, it'll stay off. If it's on, it'll stay on. And you don't have to exert. Once you've flipped it, it doesn't. Well, in biochemistry, it's like that too. And what actually happens is as you add cyclin B, the system doesn't turn on, but then suddenly it turns on by a sort of rather unknown mechanism. And now if you lower it again, you find that actually we stay in that state until we go below some threshold and it turns it turns off. This would be what the doorbell looked, very sharp threshold, but if you go back, it goes back by the same route. So the state of the switch depends on the history of the switch. So now, I said that these enzymes work by phosphorylating target proteins, and that's what makes the cell enter mitosis. So how many substrates and how much phosphorylation, and what are the sites? Well, you know, this took us a long time to figure out, and actually it turns out there are hundreds of substrates. And you can ask the question now, do they all have to be phosphorylated, or is it enough that some of them are? And of those that get phosphorylated, do they have to be 100% phosphorylation, or would 50% be enough? What about the multiply phosphorylated proteins? Do they, you know, I mean, these are difficult questions to answer. And then, if the mitotic state is with all these hundreds of proteins phosphorylated, to get out back into interphase, you have to take the phosphates off. And somebody must do this, and this was very controversial. And as a well-trained biochemist, one of the earliest lessons you learn is that there are things called futile cycles. You don't want to have the kinase and the phosphatase on at the same time, because if this is the case, then as soon as you stick on the phosphate, it comes right off again. So you never go anywhere. And all that happens is you heat up. It's just, I mean, this is terribly familiar. It's nothing, not difficult to understand. If you want to fill your wash basin in the morning with water, you must turn on the tap. That's the kinase. But it's not enough. You've got to put a plug in. Very simple. So I always thought that probably the plug, i.e. the phosphatases, must be regulated too. But there was really no evidence of that until Satoru came along, and we started doing experiments, and we realized that actually there was something wrong with the model. If you read the textbook models, they say that what's happening really in the cell is that there is this background phosphatase activity. That's the activity that will get you back into interphase. But it's just overwhelmed by kinase, because suddenly you take the Wee1 brakes off, and this huge burst of kinase activity gets you into mitosis. So we measured, here's kinase activity. You can see it actually doesn't look like that. It looks much more like this. Of course, so we thought, oh, well, it rather looks as though actually a phosphatase is being turned off in mitosis, and that's what allows the kinase to win, because the plug goes in. So we measured the phosphatase activity, and to our amazement and delight, indeed, the mitotic phosphatase suddenly switches off in M phase. This is a cell-free system. You can see the cyclin accumulating, and you can tell that we are entering mitosis by this shift in the thing, and the phosphatase comes off, and then it comes back on again. So who is this phosphatase, and what's regulating it? Well, after about 3 or 4 years' work, we discovered the phosphatase was this chap here. It's called PP2A B55. It's not a very catchy name. What's even worse is a B56 and B prime, and there's ghastly nomenclature. So here's the catalytic subunit, and the active site, I think, is in there. This thing is kind of determining what sort of substrates this guy is going to recognize, and this is just a sort of platform to align these two subunits. So now how is it switched on and off? Well, the first thing we noticed was that the B subunit had a nice juicy phosphorylation site on, but when we looked, we couldn't see any phosphate going in mitosis, so that didn't seem right. So we thought it must be something like this. Some PP1, another closely related enzyme, was known to be inhibited by a thing, I rather like the name, phosphorylatable phosphatase inhibitor. Okay, so it's a little protein, heat-stable protein, and when that binds in here, it binds much too tightly, so the enzyme can't work properly, and it just gums up the works and gets in the way of proper substrates. And we looked for this, and we couldn't find it, and we couldn't find it, and we couldn't find it. And then, as so often happens, it turned out the answer was to read the literature. And we read the literature, and the first paper we ignored completely. Great wall kinase, a nuclear protein required for proper chromosome condensation and mitotic progression in Drosophila, a fly. What's that got to do with frogs? Anyway, it's genetics, not biochemistry. But then the same group said great wall kinase participates in the CDC2 autoregulatory loop in Xenopus egg extracts. That's much too close to home. Why is this enzyme called great wall? for no other reason that the guy who worked on it, the graduate student worked on it, was Chinese. And he wanted to leave his Chinese mark on the thing. Okay, so how does Great Wall work? Well, the next thing I did was to go to a meeting, and I ran into a very nice poster by Ana Castro, and she said that Great Wall maintains mitosis through regulation of PP2A. So Great Wall is the key to turning off PP2A. What is the substrate of Great Wall? Well, again, this was, I think, an amazing tour de force by Satoru, but we discovered that this thing was called either ARP19, I forget what ARP stands for, or endosulfine, ENSA, which are little heat-stable proteins with a Great Wall phosphorylation site there. And indeed, when you phosphorylate that site, they became a potent and very specific inhibitor of this phosphatase. So that was the answer. You turn on Great Wall, it phosphorylates this thing, that inhibits the phosphatase. Is that important? Yes, it is. I apologize for this slide. This is really, you can see what beautiful data that Satoru is, an absolute technical masterpiece, measuring lots of different things. And basically, look up here. Here's cyclin accumulating. Here's our marker protein getting phosphorylated. You can see it going to 2 cycles of mitosis. And the kinase activity bounces up and down. The phosphatase activity switches off in M phase. If you take away the ENSA by an antibody depletion, what happens is the kinase activity turns on all right, but even though the kinase activity is on full blast, you have not inhibited the phosphatase, and the result is that the kinase simply can't, as fast as the kinase puts the phosphates on, the phosphatase just takes them off again. So you can never get enough phosphorylation to enter mitosis. So the story is really true. And here is a simplified system where we just have the right amount of kinase fighting with the right amount of phosphatase. And you can see this would be what would happen. This is how much activity we should get if the kinase is really on and the phosphatase is off. But actually, even though we increase the amount of kinase in the presence of a fixed amount of phosphatase, you can never turn this system off. And so the answer is, if you add then some Great Wall and endosulfine, then doing exactly the same experiment, you add a little bit of CDK1 and the system doesn't turn on, but then a little bit more, and now you begin to turn off the phosphatase, and you see this really replicates beautifully what happens in the cell. So it's not just there's no WEE1 or CDC25. This is another mechanism which regulates entry into mitosis. So this is very satisfactory, and actually these experiments were done by Satoru in collaboration with Baylor, and it was absolutely beautiful to see the match between the theory and the prediction. And I won't show you all the data, but it's really quite impressive. So the real circuit looks something like this. It actually has 2 positive feedback loops, because this, I've already referred to the mutual dependency of CDK1 and WEE1, but down here in this, here's the Great Wall bit, and because Great Wall turns off PP2A and PP2A turns off Great Wall, this is also, there are 2 positive feedback loops, and then it gets even more complicated. And in order to really explain this, you really do need these theoreticians, and this is where I begin to admire the people with their differential equations very much, because when you look at it's really quite a simple looking circuit, but figuring out how it's going to behave in real life is really tricky. And then it turns out that this module is not only used for governing entry into mitosis in these frog extracts, but also is concerned with size control in fission yeast, and it's concerned with delaying the onset of cell division so that the chromosomes get far enough apart. You don't actually, although you've turned off the kinase, the reactivation of the phosphatase is a bit slow. So Francis Barr, who discovered this, calls this the egg timer. So, the motto of all of this is that theory is all very well, but you need experiments as well. And what I've learned in my retirement, so to speak, is that experiments are all very well, but you also need theory. And it's really good to have a dialogue between the two. And I think I better stop there because I've gone on for too long. Thanks for listening.